Welcome once again. Good afternoon, good morning, good evening, and happy Halloween to you all. My name is Edgar Napoleon Asimwe. You're welcome to the 5th webinar of HOLICARE Academy and HOLICARE project. We have 3 speakers today who will be talking about microfluidics. We have Professor Aman Russom, Professor Zulfiqur Ali, and Professor Konstantinos. They will introduce themselves. I will hand over to Aman. During the webinar, you will see a pop-up for polls. We would like to receive your feedback, so please provide feedback at the beginning and at the end of the discussions. We will also share information about the project in the chats. Look out on the latest information on what HOLICARE project is doing. Allow me now at this point to welcome Professor Aman Russom. Yes.
Okay, thank you, Edgar, for the introduction. Again, my name is Aman Russom. I'm a professor at KTH, Royal Institute of Technology, Stockholm. I'm also have had the honor to be the coordinator of this HOLICARE consortium. And today's webinar is I'm going to talk about introduction to microfluidics for point-of-care diagnostics. Here is my outline. And while I'm at it, I'd like you to take the opportunity to go into menti.com and use the code that I just that's written here. I will get back to it, but briefly, I will give you a brief introduction to the HOLICARE project, and then go back to microfluidics, and then finalize this with the point-of-care diagnostics at resource-limiting settings. After me, Professor Zulf and Kostas will be able to give you more detailed around the point of care. All right, so the HOLICARE project, I will just briefly go through the motivation behind it and also finish up with the academy. What you're looking at here is a picture. This is from a review from 2014 showing the antimicrobial resistance and the threat it poses. By 2050, it's expected to be leading to the highest mortality, surpassing cancer. And here, what I'm showing you is hazards reported to WHO between 2001 and 2013. And here, again, infectious diseases are the highest. And again, when you look at the world map, you can see that the highest concentration of infectious diseases is at low resource settings. This is from a Lancet, where they basically could pinpoint that up to around 50% of the world population lacks access to diagnostics. And of course, although it was limited to few diseases, it gives you the highlight in terms of this problem is globally, and around 20% of people living in low income countries do not have access to simplest diagnostic tests at the primary care. If you look at the details, you can really see that diagnostics at the so-called the last mile particularly affects poor and rural areas, meaning that equity in terms of diagnostics would gain a lot by developing technologies that can address rural areas. If you look at respiratory tract infections, then I first would like to show you this global picture from, this is basically from 1990 to 2019. 30 years later, dominant is cardiovascular diseases and cancer. But when you look at resource-limiting settings, infectious diseases continue to dominate. It has not changed so much over the last 30 years. And lower tract infections rank greatest among the top diseases. And this is also what the consortium has been focused on, this HOLICARE consortium. Basically, HOLICARE is focused at lower tract infections because it is representing about 40% of death and 70% of cases reported in Sub-Saharan Africa. And how do we do this? Well, we are a consortium of 14 members. Among those 14 members, we have 4 from Sub-Saharan African countries, from 3 countries, actually, Ethiopia, Uganda, and Senegal. And we are addressing this holistically. And the question really is, how do you improve disease management at rural area or technically underdeveloped geographical locations? We basically combined state-of-the-art technologies, but complement this with capacity building. And this we do through a holistic approach where we have 4 pillars in parallel. One of the pillars is pillar D, which is the capacity building, and it's through this one that we are here today. And this HOLICARE Academy is aimed at human capacity building through sustainable training. So this is just to give you a little bit of background why we are here today. And let me go back then to my main topic. This is to give you an introduction to microfluidics and point-of-care diagnostics at resource-limiting settings. Again, so this is what I would like you to do. Please go into this menti.com and use the code 14232271, because I do have one question I would like to pose there. And the question is, what are we looking at here? So we are looking at a picture, and I'd like you to go online and start writing your answer, please. You will get 30 seconds. So go to menti.com, use the code 14232271. Keep writing, those that have not. So this is actually quite accurate. Most of you got it right. This is the first computer ever. And do you know what year it was? This is a question for you to keep in mind. This was in 1953, the first computer. And then 50 years later, we have the smartphones. And as we say, the rest is history. And today we can actually do sequencing at the palm of our hands. What I'm showing here is the Oxford Nanopore technology that is a really high-end sequencing machine. All this development in our field has been inspired by the development integrated circuit technology. And those who are active in the field, we sometimes say MEMS. And let me show you what MEMS stands for. MEMS stands for micro electromechanical systems. This is a technology that allows you to really take a macro scale into miniaturized world. So you can fabricate systems, methods into tiny, tiny small chips using this technology. To make things a bit more complicated, different vocabularies are used in different part of the world. Here in Europe, we will call it microsystem technology. In Japan, micro machines. In the US, MEMS. But they are referring to the same thing. So the question then is, why MEMS? Of course, improve performance, sensitivity, efficiency compared to macro scale equivalent. So with miniaturization, you can go get something that's much more a better performance, basically. The second one is that you can there's an economical incentive by doing the same thing smaller and smaller. You can have lower costs. The third thing that really resonates quite well with people who are in academia that this is quite interdisciplinary. You can come from biology, from physics, from electrical engineering, mechanical engineering, etc., and then find this no man's land where people can collaborate from their own specialties. There's a lot of applications when it comes to MEMS, everything from automobile to communication industry. When we talk about MEMS, we talk about biomems, and this is really the application we're looking at. So bio stands for biology, so biological applications of the MEMS. And the microfluidics is then used to describe things on a micro scale. This is a technology for designing, fabricating processes, and handling minute amount of liquid. Most of the time, fluids, we're talking about body fluids. And when you look at it from a development, this what we call lab on a chip concept, I will get back to what that concept is, that in the 70s, 80s, we started to think of portability. And then really in the 90s, this miniaturized fabrication technology started to make a big push. And the 20s forward, we can then see that microfluidics lab on a chip concept has been commercialized into biological applications. So what is lab on a chip then? Again, I will get back to it. So in the microelectronics, you can see that we've been able to miniaturize this large scale radios, TVs into really, really small scale chips. And really the microfluidics is trying to do the same thing with biological laboratories. So from this big concept into something that you can hold in your hand. And conceptually, this is a picture we've been showing for many, many years. So if you take a laboratory, including the people working on it, shrink it into a credit card size chip where you can then get sample in and answer out. So you integrate all the assays that you have. It could be, for instance, for infectious diseases, you might have this bacteria in your blood for sepsis, and then you would isolate nucleic acid amplification and then detection. So all will happen and then in this chip. You gain a lot, this automation, it's contamination-free integration, etc. But also there are some challenges, such as when you miniaturize your world, the volumes are inherently quite low, and then physics changes, such as, for instance, mixing becomes very, very difficult. So again, how do fluid behave on a micro scale? So when you shrink your world, what happens then is that the fluid behavior changes, such as instead of, for instance, gravitational forces, surface tension, and other type of forces starts to dominate. And characteristics of microfluidics normally is that flow is laminar, which means that the fluid goes side by side in a sheet. And to make things a bit more complicated, we also talk about different type of fluids. So when we talk about water, it's a typical Newtonian liquid. And then there's something called non-Newtonian. Think of ketchup or blood. So remember, if you try to pour out ketchup in the beginning, it doesn't flow. But when you mix it a lot, it starts to flow again. This is because it's a non-Newtonian liquid. If there one equation would love to show, or at least have to show when we deal with microfluidics, this is one dimensionless number called Reynolds number. And Reynolds number gives you a ratio between inertial forces and viscous forces, which is basically a way to explain how your system is. And because of Reynolds number typically are quite low, the flow regime are inherently laminar in microfluidics. It does not mean that you cannot do turbulent. It's just that extremely difficult because the characteristics, diameter, and also the flow rates are relatively low. So which means that typically in microfluidics, we talk about laminar. This is a very short crash course of microfluidics. So now let me go back to the lab on a chip concept. Here, really, the work is basically around how do you miniaturize, how do you integrate different processes to do biological applications. And in our field, we talk about something called point-of-care diagnostics. And point-of-care diagnostics is typically a test that could be done by anyone, anywhere, at any time. So when you do COVID tests at home, this is a typical point-of-care test where you yourself as an end user can perform your analysis. From a definition point of view, it's a testing that is defined as testing that is performed near or outside of the patient, with the result leading to possible change in the care of the patient from a definition point of view. And what point-of-care really gives you then is portability, speed, quality, and convenience as compared to doing the analysis at the laboratory. Okay, so now the second question I have for you is this. What's the first thing that you think when you hear the word point-of-care diagnostics and resource-limiting settings? You get 30 seconds to respond to this one again. So what's the first thing that you think of when you hear the word point-of-care diagnostics and resource-limiting settings? The same code, easy of use, portable, small, cheap, blood, pregnancy test. Yes, so I would say all of you are right here. And portable, again, I'm going to move forward. If you heard me talk, this is a homework of how many of you have been listening to me now. What percent of the world population globally does not have access to diagnostics? So do you remember I showed you that the global burden, the gap? Excellent. Yes, and the global picture was 47%. And then when we talk about resource limiting settings, the bare primary care systems, about 20%, 19%. So whoever wrote 47 has been listening excellently. All right, I'm going to move. So in this context, we believe that the work we do in microfluidics can address something that is crucially, for instance, infectious disease of poverty affects the poorest population in the world. Estimate about 90% of total impact of disease of poverty occurs in sub-Saharan Africa. So we believe then that microfluidics can actually aid in terms of ruling who should be treated, who should not be treated. The reason basically I'm writing this one is that most of the time, if you go to a doctor, sometimes people or the parents would more or less force the doctor to give them antibiotics to treat their children maybe, although antibiotics is not needed. So if you can have a quick test at the point of care that could guide you, oh, this patient would really need antibiotics, or no, this patient could go home, drink water and take it easy. Here, you can have a big impact in terms of who should be treated and who should not be treated. The issue then, of course, is that whenever you take, for instance, blood, you go into the central labs, you take sampling, transportation, separation, incubation, all those things are important steps that you do not have infrastructure to in resource-limiting settings. So in a nutshell, some of the barriers that we face when we talk about resource-limiting settings, that from collection, transportation, analysis, and then result coming back could take weeks to months. So this is really under this type of constraints we're talking about. And in our field, we talk about assured criteria. So when you want to develop technologies that are meant to meet the needs at resource limiting settings, we have this assured that stands for affordable, sensitive, specific, user-friendly, rapid, equipment-free, and deliverable. This is something that was coined about 20 years ago to guide, or let's say harmonize, tech developments trying to look into addressing this issue. And very recently, this was updated to with 2 letters, reassured. Reassured now has this real-time connectivity, ease of specimen collection added to that. So if you look at hospital settings globally, you talk about primary care, secondary care, and tertiary care. And at the primary care, you do something that does not require advanced equipment, but then you're limited to functionalities. And then the higher you go up into the system, the more equipment you have, the more advanced system you have. So the work we do in microfluidics for point-of-care diagnostics, trying to develop technologies that can take things that you can only afford at tertiary care into the primary care. So this is really what it is about. So I hope you get the context in terms of what we're trying to do when we say development of point-of-care diagnostics to address, let's say, less equipped areas in the primary care settings. So to give you a summary, we believe that microfluidics and point-of-care diagnostics can play a crucial role in terms of addressing the need at resource-limiting settings. And one crucial thing that we believe the technology can do is actually guide who should be treated and who should not be treated. So with that, I'm going to be giving the floor to my colleagues, and I'll be really happy to take questions and interact with you after their talk. Thank you for your attention.
Okay, well, thank you very much, Aman. So hello, everybody. I'm Zulfiqur Ali. I'll share my screen. So Edgar, if you can just confirm that you're seeing the screen.
Yes, we can see your screen.
Great, thank you very much. And thank you to Aman for introducing the point of care diagnostics and microfluidics. So I'm going to talk a little bit more around the sensor element and the fabrication approaches for these point of care diagnostics. But just, sort of start off just by reinforcing some of the things that Aman said around the need for point of care diagnostics, aspects around biomarkers of disease, and how we recognize those biomarkers through a combination of bioreceptors, and also the detection elements. And primarily that's going to be around electrochemical and optical. And then some insights in terms of how we start to make these types of devices, or some approaches for that. So as Aman says, there's a lack of resources in low, medium income countries. Also, within industrialized countries, there's aging populations. So in both cases, there's a real demand. And we can think of that in the context of chronic diseases, cancer, metabolic, or other diseases, cardiometabolic and infectious diseases. What is important is that we need to try to personalize that monitoring. And some key things, some big points is that we firstly want to miniaturize and reduce the cost of these tools. So those are the motivating factors. The other thing that is really potentially interesting is how we can make these diagnostic devices as a network of connected objects that can transmit information of the internet. And I think that provides a lot of value in different contexts. So if we think about those biomarkers of disease, And this is the FDA classification of these types of biomarkers that can be used for a variety of uses. So susceptibility biomarkers, these are biomarkers where indicators of disease before the symptoms appear. And of course, if you can do that, then there's more chance in terms of protecting from that disease. There are diagnostic biomarkers that identify and diagnose the disease as response biomarkers, and they confirm a biological response. So monitoring glucose after you've had an injection of insulin. There's other types of prognostic biomarkers. So they're indicating where a disease may reoccur or progression of disease, and then monitoring and predictive biomarkers, safety biomarkers, sort of those luckily sort of in terms of indicate extent of toxicity from a particular exposure or intervention. So bilirubin would be a safety biomarker for medications that might affect liver, or creatinine in terms of the kidney. So those are the things that we want to identify. So how do we go about identifying those? These are different types of bioreceptors that are typically used. So nanobodies are really interesting. They're about a tenth of the weight of antibodies. And what that means is that you can have a higher receptor density for these, and then potentially the higher sensitivity that might arise for your diagnostic. The enzymes and antibodies are the more commonly used receptors. There's increasing use now of molecular imprinted polymers. So this is a 3D polymer network, which is complementary to the analyte. So the analyte can fit into that. And you can modify them so that you can get reversible sensing, which can be very important. And then lastly, another a bioreceptor that's interesting is aptamers. So these are single-stranded DNA or RNA molecules. The synthetic bioreceptors, and they can have high specificity and selectivity, and they can also be synthesized at a low cost with minimal batch-to-batch variation. So that gives aptamers a good deal of advantages. There are also potential for the use of metal organic frameworks, MOFs. So these can be built up layer by layer. They tend to be porous. And what that means is that they offer a high number of sites for receptor binding, and so they can be used to enhance that sensor response. So those could be used either in the lateral flow test or on the lab on chip concept that Aman talked about earlier on. So primarily in the lateral flow test, we're recognizing that test through a visual. In the lab on chip, there's some sort of detector that we're using. So in the most common type of detectors, as I mentioned, are either electrochemical or optical. So I'll say a little bit about those. There are a wide range of electrochemical sensing approaches. They're best classified by looking at what is the control parameter and what is the measured parameter. So in potentiometry, we're essentially controlling or ensuring that no current flows through, and we're measuring the potential as a function of the concentration of the biomarker or the analyte. And these are known as equilibrium measurements because no current is flowing through. In non-equilibrium measurements, we're controlling the potential, and by controlling the potential, we allow current to flow, and we measure the current as a function of the potential. So there's a whole range of techniques, voltammetry techniques that use that. There's another commonly used technique, ampermetric analysis, and here we're controlling the potential and we're measuring the current as a function of time. So intermediate between these equilibrium and non-equilibrium measurements are the quasi-equilibrium measurements. So here, what we do is we apply an AC potential. So we apply a positive potential, we allow a small amount of current to flow, and then we reverse that potential, and so the current flows in the reverse direction. So we've got this quasi-equilibrium state. So here, essentially, what we're doing is we're measuring the time as a function, the time dependence current, and we're using that to calculate the impedance. So just to indicate that a little bit more, in ampermetric sensors, and this is one of the most commonly used sensors for glucose for diabetics, as an example, here we're using an enzyme, glucose oxidase. So that selectively converts the glucose to gluconic acid. So you've got this oxidation process. The enzyme goes from the oxidized to the reduced form, and you've got different types of mediators that can be used. But essentially, the glucose is related to the loss of electrons, or the flow of electrons into this electrode. So the glucose is related to this current. And this is the sort of plot that you get. So as you apply a particular potential, you get current. And what you'll notice here is that, you're measuring current as potential time. The current starts high and then it goes down. Initially, what you have is a combination of a charging current, which is just a function of applying that potential. It's a reorientation of the ions that are present there. So it's that charging current plus the redox current, the current that arises from this redox process. So what actually happens is that the charging current disappears very quickly. And so that's why in ampermetric analysis, we measure the current after some period of time, because we allow the capacitive current to decrease, and then we just measure the Faradaic current, or the current associated with the redox process. So the other type of sensors that I talked about are the potentiometric sensors. So here we have an ion selective membrane that's selective in this case for potassium, and as that potassium concentration increases, then the potential also increases. In the voltammetric sensors, we're applying a potential on the electrode, and we're measuring the current that results. So as the concentration of the biomarker increases, then that current will increase. So here, essentially, we're applying a potential, but that potential isn't sufficient to allow that oxidation process to take place. After we apply a certain potential, then that oxidation will arise, and as I said, the signal will increase as the concentration of the analyte increases. So the commonly type of techniques that I've mentioned previously in the voltammetry, one of these is a cyclic voltammetry. So here we have a triangular form of the potential that's applied. So the potential goes from a more negative to a positive, and then you get the reverse of that potential. So here, when we're making that potential more negative, you see that there's no Faradaic current, it's just this capacitive current. And then a small amount of that current will arise due to that redox process. And then as you increase the potential further, then this Faradaic current starts to increase. The current, what will happen at some potential though, is that all of the species will be oxidized, and what you need is that species to diffuse from the bulk to the electrode. So during this part of this classical plot, what you see is that the current will actually decrease because it's diffusion limited. And at this point, we're then going through this reverse process of where we see the reduction cycle. So really, to discriminate between the capacitive and the Faradaic current, you have a series of pulse voltammetry techniques where you have a staircase pulse and on a staircase potential that's applied, and onto that you apply a particular pulse. So in square wave pulse voltammetry, we apply this pulse, and you measure after that pulse is applied, you measure at the end. So we first of all have the anodic process and then the cathodic process. Now, both of those processes, IF, IR, will have a capacitive current attached to it. And if we carry out the IF minus the IR, then essentially you take away the capacitive current. And so these pulse voltammetry techniques tend to be more sensitive than cyclic voltammetry. The differential pulse voltammetry is just a variation on the square wave pulse voltammetry. Okay, so just turning now to optical sensors, the commonly used are colorimetric or fluorometric sensing. So in colorimetric, we have a chromophore that absorbs light, and you go to a higher energy level, and so absorption of light is dependent upon that concentration of the unlight species. In the fluorometric detection, what you have is that you have a light that excites the fluorophore. It goes to a higher energy level. You have a process known as vibrational relaxation. So that excited fluorophore bounces off against other species around it, gives off some of that energy. It goes to the lowest excited state, and there it releases a photon of light, and that's the fluorescence process. And that's shown here. So there's an excitation of the fluorophore. There's the fluorescence that arises. But what can happen is that if your anlight interacts with that excited fluorophore, then the fluorescence can be quenched. So in that case, here's the fluorescence, and when the concentration increases of your anlight, then that the fluorescence signal decreases, and that's described by the Stern-Volmer equation. Oops. So these are those three different types of receptors that we talked about: the protein receptors, the nucleic acid receptors, synthetic receptors. There are different types of interactions that can take place. So here you have the anlight binding onto the antibody, the anlight displacing a tag. Here, as an example, with the aptamer, you have the nucleic acid, single-stranded nucleic acid with a tag, and as the anlight binds to it, it changes that. You also have the anlight can cause this to swing, so it acts as a molecular pendulum. In the synthetic receptors, the anlight can bind into the MIP and change the properties. So just to describe that a little bit further, this is a good example. Here, the aptamer is bound onto the electrode. The anlight causes that single-stranded DNA, RNA to fold over on itself, and there's an electrochemical tag, which brings that tag closer to the electrode. So essentially, when you have the anlight, then as that anlight concentration increases, then you'll get an increase in the signal. Similarly, in the MIPS, if the anlight takes up the area within the MIP. It prevents the tag from taking up the space, and so that concentration decreases. What you can also have is you can have the situation where you need to have reversible sensing, and there are different types of strategies that you can use to reverse the binding of an analyte onto the surface. So that could be a competitor molecule, it could be heat, light, electrostatic repulsion, etc. So just briefly then talking about the methods of fabrication. In terms of prototyping devices, casting is the most common approach. And so what you would do is you have a photoresist that you put onto a wafer, you spin it, and if you spin it at a particular speed, you create a particular thickness, you bake that, so you raise the temperature. You can then get a mask and expose that to UV light, and where the photoresist is exposed to the UV light, in this case, it'll harden that area, and then you can develop it and remove those areas that are not hardened. That provides you with the mold. You can then cast PDMS onto the mold and get the reverse of that structure there. What you often then need to do is to place a lid onto that, and you can treat that surface with plasma or some other means to bond that onto the lid. And if you can carry out that structuring, if you can create some structure, you can do some interesting things. You can use, so here's some structures that have been created, and you can use this to control flow. So here we show that you can create vertical and horizontal valves, and this is the architecture that's commonly used. So you have a PDMS membrane, you have a control layer, which is a space there to apply a vacuum. And so if you do apply a vacuum, that PDMS membrane is deflected and it allows the flow to pass through. So in this configuration, you have got a fluidic layer that has got some architecture. You've got the PDMS membrane that is deflected, and you've got a control layer, again, some architecture where you can apply the volume, the vacuum rather. So this is another approach that you can use, which is laser cutting and joining. So here you see a number of different layers. You apply your liquid as your inlet. Aman talked about that in microfluidics at small dimensions, you get different types of behavior. And so here, what happens is the liquid flows through here, passes along here, and then it goes through the different layers and folds. And there you see a map of the London Underground with that. Those are for prototyping, low volume approaches. Hot embossing and micro-injection molding are for creating much larger volumes of devices. In the hot embossing, you have a polymer, you have a mold, you heat the polymer up, you press the mold into the polymer, and it leaves an impression of the mold. In injection molding, you have a mold, you close the movable part onto the fixed part, you have a molten polymer, you inject that into the cavity, and then you cool that down, and it leaves, again, an impression of the mold. So these are very much volume manufacturing techniques. Even higher volume are roll-to-roll type processes. So I talked about the lithographic type processes for casting. Well, you can implement those in a roll-to-roll format. So here's the exposure of a laminated dry film of photoresist. And then you can go through the process of development, and you can do a whole range of other types of processes of electroplating a metal layer, of removal, developing and you can create metal structures. So you can create the structures for electrochemical sensing. So with that, I'll thank you very much. Those are my contact details, and I'll hand you over to my colleague, Kostas.
So good afternoon to everyone. My name is Konstantinos Mitsakakis. I'm working as a group leader in immunoassays at the bioanalysis division of Hahn-Schickard and in the laboratory for MEMS applications at IMTech, the Department of Microsystems Engineering at the University of Freiburg in Germany. And the topic of my presentation is the centrifugal microfluidics for global health diagnostics. So just to position the project HOLICARE in this webinar, when we were building the proposal, we tried to handle or to tackle African healthcare challenges by means of the 4 pillars that also Aman presented in the beginning. So as to demonstrate that diagnostics is really important for those challenges, but it has to be coupled together with other activities such as capacity building, adoption and implementation, and human capital potential. The following slides are focusing on the innovative diagnostics and the pillar A. In particular, the global health and infectious disease diagnosis. So pathogen identification through nucleic acid amplification, either PCR or isothermal, is key for infectious disease evidence-based diagnosis and patient management. For technology developers like us, this means bring the technology into the chip, like lab on a chip, as was mentioned before, and bring it at the point where the care is given. That's the point of care that was also mentioned before. This means that we, or the technology developers, we need to integrate and automate the laboratory operations in order to reduce labor costs, reduce manual errors, minimize the hands-on work and time to result, make the diagnostic tools available to limited trained personnel, and save the time of the personnel to do other crucial tasks while the test is running. So microfluidics, as was mentioned before, can be a driver to such integration. And among the several microfluidic platforms available, centrifugal microfluidics is a good candidate. Why is that? Because they offer a broad palette of physical principles for liquid handling, such as centrifugal forces, capillary forces, pneumatic operations. They're modular and easily adaptable to diverse application needs, and they can be applicable for a wide range of assays, for example, nucleic acid, protein, and cell-based analysis. Typically, how these fluidics are developed is you have a partner, or you yourself, develop an assay, and you have a block diagram like this with the steps of the assay, and then there are specific unit operations, as we call them, microfluidic unit operations, each of them simulating one of these steps, and at the end, all these unit operations are interfaced into a final centrifugal microfluidic disk. And one example of them looks like this, the lab disk, a fully integrated centrifugal microfluidic platform with all the reagents pre-stored. Because remember, you need to integrate the whole lab into a chip or a disk. So for detecting, let's say, pathogen, you need to lyse them, you need to process the DNA, you need to amplify. So all these reagents need to be included in such a disk. A short video to show how a couple of such operations of the lab disk. Here you will see some liquid mixed with magnetic particles. Then the disk starts to spin. There is some mixing of the bits, and then you can imagine one magnet collecting them, and the second magnet pulling them out, transferring them to the next chamber for the purification of DNA. Then there's a purified liquid, let's say, containing DNA, and then this is by centrifugation aliquoted, metered and aliquoted to reaction chambers, where you can imagine in each one of these you have pre-stored primers, and then let's say with a specific instrument processing the disk, you can also have the real-time amplification on that device. And that device would look like this roughly. That's what we were calling the lab display 1, first generation. That was a functional model. It was quite compact, and it consisted of hardware like mechanical unit, of course, for the rotation, thermal unit for the thermocycling or isothermal, detection for fluorescence or chemiluminescence or imaging, as well as built-in magnets for the processing of magnetic particles. So this brief introduction about the lab disk. Now let's go to the implementation of such a system into the context of global health, which is the title of the presentation anyway. So we will start with the tropical infections and with a scenario. So let's assume that there's a patient who presents with acute fever at resource-limited settings. By default, malaria is assumed and the test is done first. Then let's assume malaria is negative. Then what happens? Would you do an additional test? That would mean additional cost from doing sequential tests. And what if they are not available, those tests? Then you would need to travel to the closest clinic with whatever closest means in such areas. Or maybe the doctor would give any drug that they have in the drawer, just acting under pressure. In addition to that, one could imagine that there are overlapping epidemics of dengue, chikungunya, and so on, which make the diagnostic landscape much more complicated. So acute fever is the same symptom in all these diseases, and the problem is that false diagnosis would lead to wrong drugs, and this would lead to people dying or developing resistances. So that triggered us in this project, Diagnostics, to try to develop a system for the differential diagnosis of acute fever via multiplex identification of pathogen, RNA, and DNA. For this, we chose disease panel, which was consisted of bacteria, parasites, and viruses. So representative were Salmonella, Typhim paratyphim, Streptococcus pneumoniae. From parasites, we selected the Plasmodium falciparum vivax malaria, responsible for the malaria. And viruses, we had three main ones, dengue, chikungunya, and Zika. So a panel which have all of them have same clinical symptoms, but they require different treatments. So important to be differentiated. That project led to the fever disk. So the design looked like this. Na1 is the inlet with 200 microliters of sample, which was blood or serum. Then lysis of the pathogens and extraction of DNA was happening with the use of buffers, which were stored in packages called stick packs, and this what is named Na2. Na3 shows magnetic particles, which would capture the magnetic beats. And remember the magnets, the built-in magnets mentioned before, they were transferring those magnetic beads along this structure Na4, and eventually the purified DNA RNA was transferred up here, which Na5 is a lyophilized pellet containing all the amplification enzymes. And then this mixture was aliquoted to the area Na6, where 12 chambers were hosting 12 sets of primers for detecting 12 targets, and the detection was done using real-time LAMP isothermal amplification. Eventually, the time to result totally from start to end was 70 to 120 minutes, depending on the assay. We did a small-scale validation in Senegal at the Institut Pasteur in Dakar, who, by the way, is also a partner in HOLICARE. Here you see Ben, one of our project partners, and here is Sheikh, also member of the HOLICARE. We sent 50 disks. The tests were done using collected samples from past epidemics, and the confirmation was done using some reference methods with, depending on the availability, either lateral flow tests or microscopy or benchtop RT-PCR or RT-LAMP. Some indicative outcomes are shown here. Here on the right, you see how we distributed the primer sets along the various amplification chambers. So let's say if we would get, we knew that if we would get a signal coming out of chamber number 9, that would mean that we detect dengue 1. So like here, for example. By the way, we splitted the 4 serotypes of dengue into 4 different chambers. If we would get a signal from chamber 10, that would mean detecting dengue 2 like here. A couple of highlights were, for example, I mean, those samples were characterized with RT-PCR before, as I said, because they were biobanked from previous epidemics. So during the prior characterization, one sample was known to be of dengue, but not known of which serotype. So we could manage to serotype it as dengue 1, since we got an amplification from chamber 9. There was a negative chikungunya sample. Here you see 8 chikungunya straight line. But in that sample, we sort of detected or discovered an additional malaria because we got signal from chambers 1 and 2, which were deriving from malaria. And similarly, in a positive chikungunya, we also discovered an additional dengue 2, which had been missed by the initial measurements because, as I said, in an epidemic, you typically screen for the epidemic pathogen. So this is a nice example of why such tools are important to detect co-infections, like in this last case. There was a second validation in Sudan with the help of our project partner MAST, who also are members of HOLICARE. A few discs were sent at the central laboratory in Khartoum for testing malaria primarily. And during the tests, one patient appeared there in real time being sick. He was tested during the measurements using a standard microscopy, was found positive, and then freshly collected sample was used in the disc, and it was confirmed with Plasmodium falciparum with a pretty fast assay time. And lastly, we wanted to demonstrate, let's say, multiple detection of bacteria. So we spiked all the 3 different bacteria at once in 200 microliters of blood, and we could detect all of them simultaneously with a pretty good limit of detection. So we're talking about malaria, dengue, chikungunya, and Zika, and where do these infections come from? So the answer is from here, from this one, from the mosquito. And there was a new project called DMC Malvec, where we had the motto that humans get sick because mosquitoes transmit. And the problem is that these vector-borne diseases are responsible for more than 17% of all the infectious diseases. And the occurrence of these diseases is not anymore located only in tropical areas, but it moves towards moderate climate zones as well. Of course, the main way to fight them is insecticides, but there are still challenges there. First, there's an increasing development of resistances to insecticides. And second, there is a lack of appropriate mosquito analysis tools. So this is where we were coming in, and we tried with the lab disk, which in this case was called the vector disk. We tried to help with by providing an automated analytical tool for all necessary levels of molecular-based information for vector management. So in this case, we tried to integrate 28 assays in a disk, which was pretty many. Outside the disk, we performed the smashing of the mosquitoes and the preparation, and then the prepared mosquitoes were inserted into the disk, and then we had again this mixing with lyophilized reagents and the multiplexed detection. In each chamber this time, we had either duplex or triplex RT-PCR compared to the singleplex RT-LAMP before. So what kind of molecular-based information would be needed for an efficient characterization of mosquitoes? First of all, to define if the mosquito is Anopheles gene in case of malaria, which is a DNA assay, or Aedes species if it's an arboviral vector, so an RNA assay. So that was achieved. In case a mosquito was infected with malaria, one would need to determine if the infection was from a Plasmodium falciparum or Vivax or Ovale or malaria, so the species of Plasmodium, which would be a DNA assay, and also the infective stage, how infective the mosquito was, which was an RNA assay, which was also done. And at the end, to assess the insecticide resistance levels, which was done through DNA and RNA assays as well. So eventually, that vector disk was the other side of the same coin of the fever disk, because as we saw, all these vector-borne diseases were also part of the human diagnostic vector disk, fever disk. Then we go to respiratory tract infections as a main candidate of global health threat. Why is that? First of all, the respiratory infections are major causes of hospitalization, either in primary care or in the emergency room. These are the main settings. Main respiratory pathogens are of viral and bacterial nature, and often diagnosis is done symptomatic, but if it's viral or bacterial, it needs a different treatment. So differentiation is needed. Otherwise, antibiotics are often given unnecessarily, like in case of viral infections, which triggers very much the antibiotic resistances. And all of us have heard how this is an increasing trend currently and also in the next years, and Aman mentioned it in the beginning as well. Also, why is a global threat? Because the past, say, 20 years, many of the epidemics that have happened, or pandemics, have been related to respiratory infections. In that context, we expanded the application of the lab disk towards the respi disk. And in this case, we thought, okay, let's try to integrate commercial reagents. What you saw before in the fever disk and vector disk were reagents developed during the project by partners. Now we said, okay, let's see if we can integrate ready-to-use commercial reagents. So we had the panel on the respi disk consisting of several viruses and bacteria, corona as well, but not the COVID-19 that was still below or before the COVID era. But we had the other types of the subtypes of corona. For a proof of principle study, we used 36 samples that we bought from QCMD, and they were procured as external quality assessment panels, EQA. We used UTM as a specimen, so the liquid and the reagents, as mentioned, were commercially available. And as reference method, we used the routine clinical diagnostic method of the University Clinic, Uniclinic Freiburg. The respi disk looked like this, not very different to the fever disk, some modifications towards optimization using some advanced unit operations. Again, you see sample inlet. This green shows the place where the stick packs were with those buffers. The area 2 was the bind wash elute and purification of nucleic acid. Number 5, the lyo pellet. Number 7, the PCR chambers. In this case, we used an RT-PCR in the respi disk. So the main outcomes were that we were testing challenging sample concentrations from the QCMD, which were typically used in commercial devices to test themselves. So the agreement between what we found positive and negative and what QCMD found positive, that's here. What we found positive and negative versus what QCMD found positive and negative was 85%. What Uniclinic found versus what QCMD found was also 85%. And the agreement between our results and the Uniclinic Freiburg was at 100%. So we were pretty fine with those results and published them also in Analyst. Now we've been talking this time about DNA, RNA. Maybe we've been talking too much is the question. Is the DNA and RNA the holy grail really of diagnostics? Remember the phrase we said at the beginning, pathogen identification through nucleic acid is a key for infectious disease evidence-based diagnosis. But the nucleic acid amplification alone may not differentiate colonization versus infection, may not differentiate cases of sepsis, for example, versus non-sepsis inflammations, or may not define disease severity. In these cases, if you have host response biomarkers, which are secreted by the body and not by the external agent, and if you detect them or quantify them in a complementary way with a pathogen ID, then that could improve diagnostic accuracy and efficiency. Great. But let's remember that the pathogen ID, to do it, you need the nucleic acid amplification, and to do a biomarker quantification, you need an immunoassay. So that means you need different instruments, which means costs, and you need different settings to do the analysis. For example, the microbiology for the pathogen ID versus the clinical chemistry to do the biomarker. This means that you receive the results from each of the two at different time frames, and this may delay the whole process. Eventually, what we were proposing is, what if there is an immunoassay that can be compatible with and integrable with nucleic acid amplification point-of-care instruments? So basically, we have one device which can handle a PCR cartridge and an immunoassay cartridge. To progress with our suggestion, we use CRP as a case study biomarker because it's broadly used for guiding antibiotic prescription. So, for example, cutoff values like are 20, so below 20, typically no prescription. Above 100 milligram per liter, yes. And in between, it's a gray zone where you need to co-assess with other clinical parameters. This is how was born the bound-free phase detection immunoassay, BFPD. And this picture shows the typical competitive format. So you have magnetic particles with coupled antibodies. You have fluorescent particles with coupled competitive antigen. And together with the sample, they are mixed, incubated. And after the incubation, the magnetic beads with whatever is bound on them is removed. And then you measure the supernatant or whatever is unbound fluorescent beads, therefore the name bound-free. We tested this with some real samples, clinical samples, quantified using our method, the CIVD ELISA kit and immunoturbidimetric assay commercial analyzer. And we were pretty happy to find a nice correlation between our assay and the immunoturbidimetry, so near 1, and also between our assay and the CIVD ELISA kit, also near 1. That was on the microtiter plate. Remember, we said a few slides before that it would be important if the same device could process a nucleic acid amplification and immunoassay. So that device could be LabDisplay-1, for example, proven to process a PCR disk, but what about the host response biomarkers? Well, for those, there is a immunodisk, which looks like that. And it's a full disk consisting of 3 structures, identical ones. So in principle, 3 samples on the same disk could be run with fully integrated reagents for sample-to-answer immunoassay. These are the stored beads, magnetic and fluorescence, and here is a liquid in a stick pack. Typically, the process is you insert your serum in chamber 1, you start spinning the disk. Number 2, the stick pack opens, releases the liquid, the buffer, which comes inwards, mixes with the sample. Then these 2, they go to number 3, they rehydrate the fluorescent beads, and the whole mixture goes to number 4, rehydrates magnetic beads, and then it's the incubation, separation, and detection that we saw before. So it's a pretty simple process. The heart, let's say, is this chamber, which we call it multi-purpose chamber, exactly because it does all these functions. And there's a secondary chamber on top of that, you can see it here maybe, which is a storage chamber, which helps to stabilize those magnetic and fluorescent beads during the drying process. We demonstrated that with CRP with a 20-minute time to answer. We had the standard care where we were below and above the main cutoff values. And we used the certified reference material by the EU with a non-concentration 41.2. With 3 batches, we measured it on average 46.9, which was pretty near. And we had a CV of less than 10%. We were also pretty satisfied with that. So that was an immunodisk on a functional model device. So then the next level of challenge was, how about integrating this assay into a commercial instrument, which was a spin diagronda. That was a device using a half disk for 3 viruses, SARS, RSV, and influenza. And the challenge was to integrate the BFPD on such a device where not really any hardware changes are possible, and also to demonstrate some sort of a throughput because it was a COVID-19 era. So remember before, this was the multi-purpose chamber, the heart of this BFPD assay. And so what if you have less degree of integration, so no stick packs, but you have a higher degree of throughput? So putting 7 such chambers into a half disk, that means 14 data points per run. The reproducibility between all those chambers was measured to be at 5.4%. And we demonstrated a sort of clinical application scenario, the chronic obstructive pulmonary disease, the COPD. And we wanted to semi-quantitatively classify real samples into concentration zones. And these zones were created by 2 cutoff values, the 20 and the 40. And after calculating the LOD at 2.3 milligram per liter, we tested 27 serum samples with a BFPD on the benchtop microtiter plate and on the Ronda. So among these 27, 22 of the cases were in agreement between the two methods. So the green ones means that they were classified into the right or into the same zone between the concentration zone between the two methods. So as a conclusion, the centrifugal microfluidics, they offer some advantages like modular strategy, multiplexing detection, full automation. They allow minimum hands-on steps, and they are suitable for limited trained personnel. They offer compactness, portability, and point-of-care use. Of course, multi-purpose, because we saw they can be applied in tropical diseases for human and vector diagnosis, in respiratory infections. They can detect DNA, RNA, proteins, different types of pathogens, and different sample matters: blood, serum, saliva, which I did not mention, swab, and mosquitoes. So eventually, I would like to express my deep acknowledgements to all my colleagues in the past years that have helped very, very much into these developments, the project partners, and of course, the funding agencies. Thank you very much.
Thank you. A big thanks to our dear presenters. Now we are going to do a Q&A. Q&A is now open. We had one question from Vianney. It has been answered by panelists. If you have a question, raise your hand and I will open the mic for you.
Just to say, Edgar, there was one point that was raised by my good friend Paul Marshall, who mentioned that in the fabrication approaches, I didn't mention 3D printing, and he's quite right to highlight that omission from my side. It's completely true that 3D printing, additive manufacturing is a really important technique. It provides, I think, an opportunity between that batch to volume manufacturing, and also integration of different types of elements. It allows potential for integration of valves, integration of metal electrode elements that you can use for electrochemical sensing. So I think a really interesting technology, and Paul's company is really doing some great work on that. So I think from my side, it was only because sort of matter of time, really. But yeah,
great.
Omission.
We have a question. I think this goes to you all. And the question is, how complicated is it to design and fabricate biomems, lab disks? Are these, let me take it again. So many posts going. How complicated is it to design and fabricate biomems, lab disk? Are these tailored depending on the needs or available off the shelf?
Maybe I can start with a comment. So it depends really on the application that a lab disk or such a device would be needed. If you really need to have a sample to answer disk for detecting pathogens, for example, this is, in my opinion, a relatively complicated situation, because when you design an area of the disk, you need to take into account what happens at the other side of the disk, let's say, and that's number one. Number two, let's remember that you need to really integrate a laboratory on such a disk, which means, as we saw in one slide, you need to have a lot of different biochemical agents. Some of them might be compatible with your plastic, some of them not. So you need to make a big thought about that. And losses of material, say adsorption and so on. This is really what we call heterogeneous integration of micro nano biosystems. And in terms of fabrication, when fabricating, let's say, a few tens for a proof of principle, it's one thing. And the other thing is if you want to upscale it towards a product, which means at least thousands to tens of thousands or hundreds of thousands. There, you really need quite some investment for infrastructure, for automation, let's say, to go from one station to the other, one manufacturing station to the other, to consider how you will integrate all those biochemical components in such a disk in your manufacturing line. So it is, yeah, several steps of complication. But again, if you're talking about this quite challenging thing of sample to answer nucleic acid amplification for pathogen detection, there might be some cartridges which may do part of the job, and then some other steps are manual. In that case, it might be easier.
Thank you, Konstas.
If Aman
we have
or
another.
would like to,
Yeah, maybe Zulf or Aman would like to comment.
Maybe I would like to just compliment about the simplicity. I think Zulf mentioned about hot embossing and injection molding, as well as he also showed this casting technology. Most of the time, at least in academia, we tend to use this material called PDMS, which is a rapid prototyping way of designing your chips. And that probably is relatively easy to start with, and we will be happy to provide any expertise as well as support if you just send us email. So if you, whoever asked, are in the academic constellation, I would say casting with PDMS is slightly straightforward. So please get in touch with us. Thank you.
Thank you, Aman. Another question, and this goes to Zulf. Where should one start when considering which bioreceptor to choose for optimal biosensing?
Yeah, that's a really great question. I mean, I think it depends really in terms of what your application is and what the biomarker is that you're looking at. And obviously, sort of different people start off on a different point. I mean, there will be some, I guess, antibodies that are relatively well characterized for sort of particular type of biomarkers, and so sort of that would be a sort of a place to start. But there are other more newer approaches for example, the molecular imprinted polymers or the aptamers that have, I think, some advantages over more conventional antibodies and enzymes. So I think they're attractive. It might also be linked in with your transducer that you want to integrate. So there's a good amount of work that's being done currently with aptamers and electrochemical transduction. So that might dictate the choice. So it's I'm afraid I haven't I haven't given a particularly a good answer on that, but it really depends upon the application that you're looking at, I think.
Thank you. And dear participants, you are very much welcome to raise your hand and ask some of the questions directly. There is another question which relates to what you've just answered, Zulf. How stable is optical biosensing?
I mean, one of the problems around optical sensing has always been that the elements, the light source or the detector, has tended to be in the past larger in format and more difficult to integrate into the lab-on-chip element, or it's quite costly. So one of the advantages of electrochemical sensing has been that if you can pattern metal, you can have very, very sort of miniaturized transducer elements. That in the past has been difficult for optical, but I think that's changing now, in terms of we are being able to integrate those elements. So I think it's more the optical components and integration of those that has been the challenge in optical sensing. So can we make those sufficiently low cost, or those elements are replaceable, to have those into a lab on chip concept. So what you could have is that you have the lab on chip, but the source and the detector could be components that are reusable, and that's I think a more appropriate approach. Or you think of very novel optical elements that are integrated that are sufficiently low cost. So I think those are different approaches. But I don't know, Aman might want to have some comment on that as well.
No, I think you got it quite right, Zulf.
Thank you, Aman. We have a question from Abu Bakr. I'm not sure what you mean, but the comment is great presentations. Thank you. What is next? If you are referring to the webinar series, we have more webinars coming up on different topics: biobanking, data connectivity, LFT. So as long as you are registered on HOLICARE Academy portal, you will be receiving information. So stay tuned. We have another question, comment from Asma Jamali. Would you like to read your question, comment, Asma? Otherwise, I will take Amity. You have your hand up. You have the flow. Amity?
Yeah, hello.
Hello?
Hello. So my question is from to Professor Zulfiqur Ali. So my question
Go
is
ahead.
Hi, Amit, and happy Diwali, by the way.
happy Diwali, Professor Zulf. How are you?
Fine, thanks.
Yeah.
So,
So my question is, what do you think are the primary challenges in scaling up microfluidic devices for real world application, and how can this be addressed?
okay, I think Kostas mentioned some of those points, and I think Hahn-Schickard are doing some great work in terms of moving that towards high volume type devices. And the sort of example that Kostas gave in the work that he's been doing, sort of a good examples around that. One of the aspects is, sort of, does that application have sufficient need for volume manufacturing? So is there sufficient demand for that manufacture? Or is the microfluidic device or lab on chip device, is it providing sufficient value? I think that largely goes in terms of, so the lab on chip could be constrained in terms of functions to perform particular functions, and it might do something very valuable that gives a very valuable clinical answer. So in that case, I think it would be, sort of okay. So generally, I would say we have a lot of understanding around technology, but it's and this relates to one of the earlier questions: is does that technology development actually provide useful clinical information? So I don't know whether Kostas and Aman want to comment on that, so I'm sure they've got valuable insights there.
Yeah, okay. Thank you, Professor Ali. So, my one more small question is that how do you see microfluidics evolving alongside other technologies like AI or biosensing platforms to improve diagnostic accuracy?
So that's a great question, Amit. And I think that AI could be particularly valuable insofar as, so for example, you have the transducer undergoes fouling, and can you recognize that? So that could be one element. The other element could be if you're getting a large amount of data, can you use AI to be able to extract useful information out of that? So I think those could be, sort of two useful applications of that. And I know that some people have started to work in that area as well. But good
Okay.
question.
Okay. Thank you, Professor Ali.
Thank you, Zulf. In the interest of time, because it's now 14:30, I will take just two questions before we can close. And the last question is, how easy or difficult is it for clinicians to use the fever disc for differential diagnosis in their daily work?
Yeah, that's a nice question. There was a question also which I answered in written. So in principle, you have a new system like the fever disc. First, you need to test it with spiked samples in laboratory settings, then with real samples in laboratory settings, and then to go out to the field. To go out to the field, it's something which needs to, you need to test the robustness, you need to test the user friendliness at sort of challenging conditions. The fever disc was still at the level 2, so laboratory settings with real samples. But I think in general, technologies of this type and those technologies also developed in HOLICARE to go to the field and be used by clinicians, they need also to undergo a user friendliness sort of study, get feedback by the end users, and maybe go through another iteration of modifications before they are deployed to the field. In any case, the user friendliness, I think it's a very crucial thing. Although I must say, in HOLICARE we have this training and capacity building. So in principle, we are also training end users to be able to do some manual processes and to be able to handle such tools. So that's why I said also in the beginning, it's a holistic approach, and that's the name of the project, to not only provide boxes of diagnostic tools, but also train the potential end users how to use them with the maximum efficiency.
Just to follow up on Kostas, if you allow me to do some advertisement, actually, in a couple of weeks, we're going to be in Mbarara, Uganda, doing a workshop where the end users will actually build a centrifugal platform and test it. So these are the type of holistic approach that Kostas is talking about.
Thank you so much. And the last question is simple. Marshall Paul, yes, this recording will be uploaded on YouTube channel, HOLICARE YouTube channel, as well as the HOLICARE Academy learning platform. At this point, I would like to thank our guest speakers for the interesting presentations, but also the participants for staying up to the end. Stay tuned for future webinars, and thank you, everyone.